Good afternoon, everyone. My name is LaDonna Wyatt, and I am dedicated to discovering an effective way to produce biofuel from cyanobacteria to reduce fossil fuel emissions into our atmosphere. A genetically engineered cyanobacterium, Formella diplosyphon, strain B481SD, was grown in the presence of nanoparticles at varying concentrations and Fourier transform ion cyclotron Renaissance mass spectrometry was performed to characterize the lipid profile of the nano-treated cyanobacteria cells by measuring the ion cyclotron frequency. Cyclotron frequency refers to the charged molecules which rotate at a frequency that is inversely proportional to its mass to charge ratio. The lighter ions are going to have a higher frequency than the bigger ones or the heavier molecules. The picture that you see to your right is a picture of the cultures that are grown in flasks that are 250 ml, and the concentration of nanoparticles into each one is 0.4, 0.8, 1.6, and 3.2 milligrams per liter. The figure to your left indicates the results of our preliminary growth study. Cultures grown in sterilized flasks and exposed to varying concentrations of nanoparticles at 0.4, 0.8, 1.6, and 3.2 milligrams per liter showed significant increase in growth. Cultures grown in the absence of nanoparticles and in the presence of FeEDTA served as the controls. After day 15, data was analyzed and it was determined that all nanoparticle treated cultures show significant increase in growth when compared to the control, allowing us to progress into polar lipid extraction. We used the online liquid chromatography coupled with ESI FTICR-MS using 21 Tesla to visualize the nanoparticles and also to see the compounds that are in the samples. Nano LC improves the electrospray ionization efficiency, and when combined with FTICR-MS can significantly extend the lipids coverage. Then we will research against lipid libraries to confirm lipid identities and use tandem mass to explore them. The picture to your right is a picture of our samples after polar lipid extraction was performed right before we shipped it to the MagLab. After completing preliminary studies, it was concluded that nanoparticles have a significantly higher impact on B481SD growth. Future studies will be aimed at further exploring the lipid content of these nano-treated cultures. Polar lipid extraction was performed but did not produce conclusive results due to chemical contamination. Moving forward, we have attained numerous new flasks for growing cultures, which we hope will prevent the reoccurrence of this issue. My current research is considered to be novel because little information is given on nanoparticles' impact on the cells for biofuel production. With the assistance of the National MagLab, I was able to explore various measurement techniques to gain more insight into nanoparticles' effectiveness on cells at various biological and chemical levels. Future studies will involve looking further into polar lipid extraction, optimizing our polar lipid extraction protocol, and then comprehensive molecular characterization of F-liposyphon lipid extracts will be conducted by FTICR-MS. I would like to give a special thanks to my mentor, Dr. Juan Chen.